Welcome to Sciencera. In this video, we're going to discuss nucleic acid structures and function. The main focus for today's video will be DNA. Before we move on, if you haven't subscribed to my channel, I would like you to request you to all subscribe to my channel. Let's begin. DNA. DNA stands for deoxyribonucleic acid, which is a molecule that contains instruction on organisms need to develop life and reproduce. These instructions are found inside every cell and are passed down from parents to their children. It is a nucleic acid and is one of the four major types of macromolecules that are known to be essential for all forms of life. DNA is found in the nucleus, with a small amount of DNA also present in mitochondria in eukaryotes. DNA structures. There are two nucleobases, purines and pyrimidines. Purines include adenine and guanine, while pyrimidines include thymine and cytosine. Purine always combined with thymine, AT, they always form a bond, adenine with thymine and guanine with cytosine. So they are known as the complementary nucleobase pair. Let's see the DNA structure in detail. DNA structure. The work of Rosalind Franklin led to Watson and Crick discovery. Franklin first had pointed out that DNA is made up of two spirals. The structure of DNA is a double helix structure because it looks like a twisted ladder. The sides of a ladder are made of alternating sugar, deoxyribose, and phosphate molecules, while the steps of the ladder are made up of the pairs of nitrogen bases. There are four types of nitrogen bases, which are adenine, thymine, guanine, cytosine, DNA pairing. The nitrogen bases have a specific pairing pattern. This pairing pattern occurs because the amount of adenine equals the amount of thymine, the amount of guanine equals the amount of cytosine. The pairs are held together by the hydrogen bond. Structure and composition of DNA. DNA is double-stranded helix. That is, each DNA molecule is comprised of two biopolymer strands coiling around each other to form a double helix structure. These two DNA strands are called polynucleotides. As they are made up of simpler monomer units called nucleotides, each strand has a 5' end with a phosphate group and 3' end with a hydroxyl group. The strands are anti-parallel, meaning that one strand runs in 5 to 3 direction while the other strand runs in 3 to 5 direction. The two strands are held together by hydrogen bonds and are complementary, and they are complementary to each other. Basically, the DNA is composed of deoxyribonucleotide. Deoxyribonucleotides are linked together by 3,5 phosphodiester bonds. The nitrogenous base that compose the deoxyribonucleotide include adenine, cytosine, thymine, and guanine. The complementary of the strands are due to the nature of nitrogenous bases. The bases adenine always, as we said before, interact with thymine on the opposite strand via two hydrogen bond. Cytosine always interact with guanine via three hydrogen bond on the opposite strand. The shape of the helix is stabilized by hydrogen bonding and hydrophobic interaction between bases. The diameter of double helix is 2 nanometer and the double helical structure repeats at an interval of 3,4 nanometer, which corresponds to 10 base pairs. Major and minor grooves on the DNA. As a result of the double helical nature of DNA, the molecule has two asymmetric grooves. One groove is smaller than the other. This asymmetry is a result of geometrical configuration of the bonds between phosphate, sugar, and base groups that force the base groups to attach at 120 degrees angle instead of 180 degrees angle. The larger groove is called the major groove, occurs when backbones are far apart, while smaller one is called the minor groove, occur when backbones are close together. Since major and minor grooves exposes the edges of the bases, the grooves can be used to tell the base sequence of the specific DNA molecule. The possibility of such recognition is critical since protein must be able to recognize specific DNA sequences on which to bind for the proper function of the body and cell to be carried out. Then we have properties of DNA. DNA helix can be right-handed or left-handed, but the B configuration of DNA having the right-handed helix is the most stable. On heating, the two strands of DNA separate from each other, and on cooling these, again they hybridize. The temperature at which the two strands separate completely is known as the melting temperature. Melting temperature is specific for each specific sequence. The B sample of the DNA having higher melting point must have more cytosine and guanine content, because cytosine and guanine pair has three hydrogen bonds. The sequence of bases along the DNA molecule encodes for sequence of amino acid in every protein in all organisms. Types of DNA. Eukaryotic organisms such as animal, plants, fungi store most of their DNA inside the cell nucleus and some of their DNA in organelles such as mitochondria. Based on the location, DNA may be nuclear DNA, which means it is located in the nucleus of eukaryote cells, usually has two copies per cell. The structure of nuclear DNA chromosome is linear with open end and includes 46 chromosomes containing 3 billion nucleotides. Nuclear DNA is diploid, ordinarily, ordinarily inheriting the DNA from two parents. The mutation rate for nuclear DNA is less than 0.3%. Then we have mitochondrial DNA. Mitochondrial DNA is the DNA in the mitochondria, contains 100 to 1000 copies per cell. Mitochondrial DNA chromosomes usually have closed circular structure and contain, for example, 16,569 nucleotides in human. Mitochondrial DNA is haploid, coming only from the mother. The mutation rate for mitochondrial DNA is generally higher than of the nuclear DNA. Then we have the forms of DNA. As you can see on the screen, DNA can occur in four forms: aDNA, bDNA, cDNA, and zDNA. There are more forms of DNA too. Most of the DNA is in the classic Watson-Crick model, simply called as bDNA or B form DNA. In certain conditions, different forms of DNAs are found to be appeared like aDNA, zDNA, cDNA, ddNA, eDNA. This deviation in forms are based on their structural diversity. Let's look at the forms of DNA. bDNA is most common, originally deduced from the X-ray diffraction of sodium salt of DNA fibers at 92% relative humidity. Then we have aDNA, originally identified by X-ray diffraction of analysis of DNA fibers at 75% relative humidity. zDNA is left-handed double helical structure, winds to the left in a zigzag pattern. cDNA formed at 66% relative humidity and in presence of lithium and magnesium ions. ddNA is a rare variant with 8 base pair per helical turn from form in structure devoid of guanine. eDNA extended or eccentric DNA. Then we have functions of DNA. DNA has a crucial role as genetic material in most living organisms. It carries genetic information from cell to cell and from generation to generation. Thus, its major functions include storing genetic information, directing protein synthesis, determining genetic code, directly responsible for metabolic activities, evolution, heredity, and differentiation. It is a stable molecule and holds more complex information for longer periods of time. Thank you for watching. If you like my video, don't forget to subscribe for more.